Hi. Yeah, so I'm Arthur. I work at Genomics England as a bioengineer in our cancer long-reads squad. So this wordy title is not very good, I'm afraid. So I'll jump straight in and discuss what our long-reads program, cancer long-reads program is, and then I'll talk about the workflows. So our long-reads program is comprised of two things. There's the long-read sequencing side, which is the ONT stuff, and then there's also the multimodal data side, which is about imaging data, but I'm not going to cover that here. That's not the project I work on. So the long-read sequencing side, so we're using Nanopore to do the sequencing, and it's happening as well at with the NHS at what they call these Genomic Laboratory Hubs, and there's going to be 7 of those around the country that are performing this. And what sequences do we have? So we've got 3 Prometheans at Hinxton Lab. That's gel, like ourselves that do that. And then there'll be 1 Promethean per NHS GLH, and from here on out, all our sequencing will happen on the R10 flow cells. So at capacity, we can do quite a lot. So what are the objectives of this program? So 1, faster diagnostics. So the idea of being able to do the sequencing in these labs, which are basically 7 hospitals around the country, so close to the patient, you have to wait for sample transfer and stuff. 2, more comprehensive data. So the idea that we can just do sequencing and get results for all of these things, whereas currently you would have to run multiple tests to get, say, methylation information and stuff. Sort of to review the landscape to make sure we're using the best tool. So, is ONT going to be the best tool moving forward versus Illumina, etc.? And then equitable access for this stuff. So the trial that we're doing at the moment, or this program at the moment, covers 4 clinical indications: AML, ALL, CNS tumors, and sarcomas. And eventually will be rolled out to these 7 GLHs. At the moment, we're just in 2 pilot sites, which we've started the last couple of months. So I'll talk about the overall workflow now, how we're processing the data. So we start out, so I've got this color code. So anything in blue is like the NHS and the labs. Green is happening in gel, but on AWS. And the stuff in pink is happening on our in-house cluster, basically. So we start out with the data coming from GLHs or from Hinxton, and that's transferred onto our HPC storage in gel. This is actually quite an onerous step at the moment. It's not very fast, but some of my colleagues have worked very hard to improve that and make that more stable. So then we land it in a specific location, and when it lands, we have processors that are monitoring for that and can trigger the pipeline from there on out. And this all happens with like a series of Lambda functions and AWS stuff. So the first thing it does is it triggers the single flow cell workflow, and I'll cover these workflows a bit more specifically after this. And that's going to process the whole flow cell's worth of data. At the end of that, we check to see whether the other flow cells for the same patient are available, whether they've already been sequenced and passed their QC. And if they have, we move on and do the multi-flow cell workflow, so the tumor normal analysis, and then the output from that is what gets returned to the clinicians. So the VCF, basically, of the variants that are relevant for this clinical indication. So that's sort of like the high level of our pipeline. And the two workflows there, which are Nextflow workflows, we're running them through Tower, through the API, triggered from AWS. So I'll just discuss the single flow cell workflow first. So this is designed to work on a single flow cell's worth of data. For us, 1 flow cell is always like 1 patient's data. We don't have like multiple samples in the same flow cell, so it's all 1 patient. So we can start from anywhere really, any like of the input data that can come off of the sequencer. So either the fast5s or the newer pod5s, or if you've done the base calling on the sequencer and have fast queues, we can start from there as well. So we do some conversion to pod5 if you give us fast5s, and then we can base call with either Dorado or Guppy. We started out with Guppy and we're transitioning to Dorado now, as that's what ONT are moving towards, but we haven't finished fully validating that yet. But we can do either as an option. And alignment happens as part of the base calling for us, and we also get the methylation information as well. If we get fast queues given to us, we do the alignment ourselves using the same Minimap2 parameters that are run in Guppy or Dorado, and then it feeds into the pipeline at that point as BAMs. We just do some simple QC stuff on the BAMs, some merging and sorting and indexing of the multiple BAMs that come from Guppy and Dorado. We run these various QC tools, and then we generate a couple of reports at the end. The QC report there is basically an internal gel format that we have that's just a big JSON with some information in it. And we also run Nanoplot on the summary report from the Guppy data, or from the sequencer. So I'll just discuss a little bit what some of those tools we're using them for. So for MosDepth, we're using that to get coverage of X and Y chromosomes, and we use that for a simplistic sex inference that goes into the report at the end. It's quite simple, really. We're using GenomeCov for the general 20x, 30x depth across the whole genome. And I think there's probably scope for us to just use one of these two tools to do both of these things. But this was how it was from the start, and we haven't combined that yet. Nanoplot, so we use that for the pre-alignment metrics just from the sequencer, so we can see like what we lost at the alignment step. And then we're using HLA-LA, which gives us HLA allele calls at the G group resolution, because, well, for two reasons. So there's some interest in it in other areas, and although this is a cancer project, GEL recently also announced that they are going to sequence 7,500 participants worth of rare disease data on the ONTs, so there's some hope that this pipeline will run on that data as well. But we are also potentially looking at using the allele calls to derive loss of heterozygosity in the tumor samples, and also at the very start of this project we were using it as a crude measure of sample consistency before we had the SMV calling, the downstream SMV calling implemented. So now I'll discuss the second one, the tumor normal workflow, which looks very short at the moment. So this works on the BAMs from the original single flow cell workflow, and it can take as many BAMs as it likes for like one patient. You could have one flow cell's worth of germline data and like 3 flow cells worth of tumor, or 2 of one and 1 of the other, and it will just merge them. So you have 1 of each, and then it will run Savannah, which is the tool we're using for SV calling on ONT data, and CLARE-S, which we're using for small nucleotide variants. We do a step to reformat the VCFs that come out of that to then pipe into our tiering step, and I'll explain what that is in a second. So yeah, so Savannah is the SV caller we're using. CLARE-S is the SMV caller. So tiering is a gel process that we have that tiers the variants relative to their, or for the relevance based on the cancer indication. So as part of this pipeline, the clinical data that we pass in is, for example, patient age, things like that, what kind of tumor it is, and that sort of thing. And that is that goes into our tiering process and helps us to determine which are the relevant variants of the ones that we find for the clinician. We also, so not in that pipeline at the moment, but coming as we validate these things, is going to be methylation analysis. So we'll add ModBAM to bed into the pipeline, although I think ModKit now is going to replace that. And then we can do promoter methylation analysis and hopefully CNS tumor classification. Also CMV calling, but we're still working on validating what we think is the best CMV caller for ONT long-read data. So we haven't added that into our production pipeline yet. And also phasing of the variants. I've actually flown through this, so I've got plenty of time for questions. So thank you to all these people. This is the team that I work with at GEL. Big thank you to Anthony at ONT, who's basically had to listen to all my complaints and answer all my emails with regards to Guppy and Dorado, and it's been fantastic. And then to everyone at NF-Core and Nextflow. And actually, since I've got a bit of time, I'll just mention about that we use NF-Core quite heavily for our pipelines, although we're not publishing them publicly. And it's really useful. I'd say it makes the job of writing pipelines, even in institutions, super quick, and it gives you more time to work on the things that aren't publicly covered. So I get to dedicate more time to the steps that are unique to GEL and that take a lot of effort. And a lot of the easy stuff is handled by NF-Core already, and that's brilliant. So a big thank you to that community, yes.
we've got a couple of questions, probably more than I can remember. So I've got the laptop here.
That's
Here we're going. Any plans to publish these pipelines publicly?
No comment. I don't know. Basically, I don't know. I don't know, and I'm not going to commit to that standing up here.
Fair enough. CNV callers that you're validating.
Again, I wouldn't like to comment on that. Tim and Rowan are our data scientists who are working on that, and I don't want to say the wrong thing, basically.
I'll give you something a little bit more general then. Long-read sequencing, where do you see that going relative to all of the kind of high-throughput, short-read stuff which is happening at GEL?
So obviously, yeah, as I said, so this pilot is pretty large, but this is focused solely on the cancer data. But we, as I say, Matt Brown, our Chief Scientific Officer, announced, I think, last month at our research summit that they're going to start this other project, which is 7,500 participants worth of rare disease data. So it's expanding a lot, but yeah, I don't think it's replacing any of the other stuff yet.
Great, thanks so much.
Cheers.